All right, so we're going to wrap up the last part of chapter 1 and start chapter 2 today. So we're not going to spend a lot more time on organization in biological systems. So we talked a little bit about cellular specialization last time. And from our perspective, where this semester we're going to be studying and focusing more on the molecules that build these complex systems. Okay, the thing that is sort of important for us to have perspective-wise is that when you get multicellular organisms that have complicated organizational systems that have different systems that have to be able to communicate with one another and speak to one another, okay, because they're going to be responding to environmental changes, okay? The entire way of having those responses, okay, happens through intermolecular communication and intramolecular. So communication between molecules is really how response happens, okay? So So the structural features and the regulatory features that we talk about are going to confer the possibility of having this type of organizational structure within an organism, okay? So even our ability to detect pressure, right? If I push down on my arm, okay, the way that I sense that pressure, okay, is through conformational changes in molecules, okay, that then transmit a signal, okay, and it can be an electrical signal, but even electrical signals involve the movement of ions when we're talking about nerve conduction, okay. So the way that molecules or systems that are farther apart from one another, communicate is through electrical conduction through nerves or through molecules diffusing and moving from one place to another and resulting in something we call signal transduction, okay? So this is really how a molecular interaction results in a response. And that response will be to some change in conditions, right? All right. So again, in biology, the highest level of organization are in ecosystems, okay, and so an organism's responses to its environment that happen kind of on the individual level, okay, can, when you add them up in communities of organisms, result in these macroscopic features that we see, okay. And so what happens, for example, on the right, you see a toxic algal bloom, okay? So what happens is change in environmental conditions. The water warms, okay? That causes the red algae to overgrow, okay? Which reduces the oxygen in the water. Other organisms sense that reduced oxygen level, and they have to respond accordingly, okay? So ecosystems are the way that individual organisms can interact with one another. Okay. All right. All right. So the end of chapter 1 is going to be a little bit of a a preview to the function of nucleic acids, which is what we're going to study after we finish chapter 2. We're going to talk about energetics and intermolecular forces a little bit more, and then we're going to start talking about DNA. But all of our cellular functions, okay, are going to be derived from the information that's in the DNA, okay? And so just to give you a little bit of perspective of historically, how long we've understood what kind of drives and directs all of the molecular interactions in a cell, okay? We didn't even know, okay, 100 years ago, we didn't even know that DNA was the thing that was controlling, what happens in cells, okay? So it wasn't until 1952, okay, that we determined that, all certain viruses need is DNA to replicate, to infect other cells and to make copies of themselves, okay? So that was when we were starting to hone in on nucleic acids as the information molecule in biological organisms, okay? What you see on the screen, okay, is Rosalind Franklin's X-ray diffraction data. This is the famous plate 51 picture, okay? And so Rosalind Franklin bombarded nucleic acid crystals of nucleic acid with X-rays, which is today, X-ray diffraction is one of the key methods for determining the structure of biological molecules. So X-ray diffraction data is a key structure determination technique. So in organic chemistry, you learned about NMR, and the type of NMR you had experience with in organic chemistry is good for, again, those molecules that have a couple of hundred atoms maybe. But when you get to biological molecules with thousands of them, you need to employ other techniques, as well as you can do NMR, but you need a much, much stronger magnet. So for example, the silver bullet we have over on the other side of the building is a 400 megahertz magnet. To do the structure of a small protein, okay, you need something like a 900 megahertz magnet. Okay, so something that's twice as strong as the one we have here. And you also need to do different techniques. You need to look at different nuclei. You need to do what we call multidimensional NMR. And there's a lot of computation that goes into interpreting the data to be able to get a structure out of it. Whereas with X-ray diffraction, from the raw data, you start to get a little bit of a picture of the structure more easily. So this is looking kind of down the axis of the DNA helix. So X-ray diffraction data helped us put together a molecular picture of DNA, that double helix. Okay, and one of the reasons that DNA is a good example to start with, okay, is that once we finally knew the structure of DNA and we had a model that said, oh yes, we have two strands and the nucleobases hydrogen bond with one another and they pair in specific patterns. So once we knew those structural rules, we were able to use that to determine how the molecule functioned. Okay, the fact that we can get information being transferred, say, from DNA to RNA, and then from RNA into protein, all lies in the nature of the base pairs, okay? And so having that structural insight about the base pairing and then the structure helped us to sort of understand function. So this is structure guiding our understanding of function. And that's a key thing to keep in mind. We're going to study these molecules in structural detail so that we can understand how they work, okay? And we can get kind of rules of how they work. Okay, so we'll go over this a lot more again when we go through chapter 3, but there are 3 basic parts to a nucleotide, okay? So there's the base that's shown in blue, and then there is a sugar entity, and it is ribose in RNA, okay, and it's deoxyribose in DNA, okay. And the difference lies in, again, just a hydroxyl group at the 2' position, or the lack of one, and that confers differing chemical stabilities on molecules, which also relate to their function. Okay. The base pairs, or the base that's attached to the sugar, okay, is going to be what's involved in base pairing. And so this is a part of the nucleotide that carries the information. And then the phosphoryl group, which we won't neglect, okay, is how we make it's involved in the linkage between nucleotide monomers to make the polymer. Okay, so required to link monomers. Okay, so the way information is going to be transmitted is through what we call complementary base pairing. So if there's a spelling error on this slide, so it should be complement with an E, not complement with an I. So complement is like, gee, I really like your sweater today. Complement means 2 things go together. Okay, so, all right. So, complementary base pairs, it means I always pair with a certain type of partner, right? So, guanine, which we abbreviate as G, always pairs with C. Oops. And adenine, which we abbreviate as an A, always pairs with thymine, which we abbreviate as a T. So this is going to be kind of our first introduction to the shorthand way of referring to monomers. Okay, so we will talk, as we talk about the polymers that we're going to study in this course, sequence, the order in which you connect different types of monomers, okay, is going to dictate all the other features of the molecule. And so communicating sequence is important. But when you have hundreds and thousands of things linked together, if you were to write out the name of everything in the order that they were connected, it would get very long, okay? So for nucleic acids and proteins, we have what are called 1-letter codes. Okay, so we have abbreviations that refer to a specific type of monomer. Okay. Oops. So we'll just call this the 1-letter code. And all that does is it helps make communicating sequence take less space. Less space. Okay, so same thing with proteins. When we get to carbohydrates, okay, we are going to have such a wide variety of monomers. We're going to have dozens and dozens of different types of monomers, and they could have multiple different types of connections, okay? And so instead of using letters, because for letters, we are limited to, 26 of them, right, in the English alphabet. So for carbohydrates, instead of having, letters like this, what we use are symbols and colors, and then we also use letters to determine, and numbers to determine linkage type, okay? So we'll talk about, you guys have learned about IUPAC naming in organic chemistry, right? Well, in carbohydrate chemistry, there is the equivalent of IUPAC. It's, a blue triangle means this, and a red circle means that. And that helps us to depict the sequences and the connections between monomers in an efficient way, so that, it fits on page or in a figure. Okay, so these base pairs that form between nucleotide monomers, okay, they're formed because of, we didn't look at it too closely here, but because of hydrogen bonding, okay, between groups that are on the bases, okay, and so with our GC base pair, we have 3 hydrogen bonds. With our AT, we have 2. Okay. Structurally, those are going to be found in the center of the helix. Okay, so when we talk about our polymers, we will talk about backbones in DNA and proteins. In carbohydrates, we'll talk about main chains. Okay, so the backbone is going to be where the polymers link together. Okay, and just like when you have Legos, you have the knobs and the holes. That's how you connect them. You stick them together. Well, when you have monomers that join to form a polymer, okay, every polymer class is going to have functional groups that the monomers have to connect together, okay? And so that means that you may have 20 different monomers or 4 different monomers in DNA, but they all have to have something chemically similar to be able to connect, okay? And once you make those connections, there's a part of the molecule, okay, that is kind of just repetitive and all the same. And then there's a part of the molecule that looks unique because of the sequence of the different monomers, okay? So this here is, oops, that's not what I wanted to do. I wanted to do that. So this right here is the backbone. And, whoops, and that winds up being physically on the exterior of the molecule. And the interior, okay, this is the sequence dependent part. And I'll put in parentheses unique part of the molecule, okay? All right. And so the fact that these are connected by hydrogen bonds are complementary base pairing, okay? Remember that hydrogen bonds are geometry dependent, okay? The distance between the functional groups, the angles at which they're being held, okay? So that's why you get the specificity, and I will repeat this again next week. You get the specificity of the base pairing because, the way the geometry of a guanine is and a cytosine it holds those functional groups in such a way that they can hydrogen bond, whereas in your normal DNA polymer, an adenine or a thymine is not going to be held in that way where it can hydrogen bond to the other thing. So you get to the guanine. So you get specificity, okay, because of geometry, okay? So hydrogen bond formation is geometry dependent. And how we get specific base pairing rules. And as we dive further into our study of nucleic acids, what we'll find is there's this what we call the standard or canonical base pairs, and then we have what are called non-canonical base pairs. So in cases where you have different geometry, okay, you can get base pairs that aren't, you can get a GT base pair or a GA base pair, okay? And they're not that uncommon, okay? But they are found not in the typical double helix that we think of when we think of DNA. They occur in transfer RNA molecules when they interact with messenger RNA, and they occur in these multi-stranded weird DNA structures that you get in certain phases of the cell cycle and cellular functions, okay? All right, but because in our standard DNA we have this rule for A always pairs with T, okay, we can make copies of the molecule using each strand as a template, okay? And so this shows you, DNA being copied from the template using those base pairing rules, okay? Okay, so the central dogma, ooh, that's awful. All right, so the central dogma, hello, that's weird. Let's go for white. It's not writing. All right, we won't circle the central dogma. So when I say central dogma, okay, the central dogma of molecular biology is the order of information transfer from DNA to final functional response or final functional molecule, okay? So what that means, okay, is we go from DNA, which is our long-term storage molecule. Let's get my handwriting legible. And that long-term storage molecule can be copied, as in replication, or it can be used, okay, that information can be mobilized to have some type of cellular response, okay? So transcription, okay, results in an RNA copy, okay, so RNA copy of DNA. Okay. And we have multiple different types of RNA. Okay, but the RNA is our information transport molecule. Okay. So this is information transport. But most of the time, okay, the RNA isn't the final molecule that's going to perform the function that's involved in responding to the environment. So that gets translated, okay, into protein. Okay, so this will be the functional response to an environmental change. or just to the environment you're existing in. Okay. So the central dogma, okay, as it is often summarized as DNA to RNA to protein. That's what we mean with the central dogma. Now, there are all sorts of variations on this theme. Okay, so in certain viruses, okay, we get what's known as reverse transcription. So, for example, the HIV virus, its DNA is stored in RNA, its information is stored in RNA form. The SARS-CoV-2 virus is an RNA virus, okay? Now, some RNA viruses, okay, in order to make copies of themselves, okay, they actually do something called reverse transcription. And so what they do is they have an enzyme that makes a DNA copy from the RNA, okay? And so this is what HIV does. So HIV goes around as an RNA virus. That's the infectious form. It has an enzyme called reverse transcriptase, okay? And that makes it, when it gets into the cell, that makes a DNA copy of the RNA genome, okay? But the type of virus that HIV is, it's a lentivirus, so what happens is that's a virus that can integrate into the host cell's chromosome. So it has another enzyme called integrase that takes that DNA copy and it plugs it into the genome of your cells, which is why HIV is, it's symptomatically treatable, but it's really super hard to cure, and because it kind of gets itself integrated into the host cells. Okay, COVID-19 is not that kind of virus. Okay, it carries different enzymes and has sort of a different mechanism of replicating itself, but it doesn't integrate into your host cells. Okay, so the proteins that get made, do all the functions of the cell. Okay, so RNA sometimes, okay, does some of the. This should have a P and an SYN there. The ribosomal and tRNA are the ones that help us make proteins, okay, along with the mRNA. But these other types of RNA are involved in processing other RNA molecules. So, all righty. Okay, so let's talk just briefly about how DNA and RNA and final protein sequence are related. You've seen this, I'm sure, in biology classes, even if you just took one in high school, right? So we've got these base pairing rules, okay, and information is stored on the DNA. It's stored in one of the strands, which we call the coding strand. The template strand is the one that we make an RNA copy of, okay? And so it actually, in sequence, it matches the coding strand, okay? So what happens is we take our two strands of DNA, they get opened up, okay, the template gets used to make a copy, an RNA copy of the coding strand. Okay, that coding strand, okay, has information stored in what we call codons. Codons are units of 3 nucleic acids, right? And each triplet, each 3-letter word codes for a different amino acid or is a signal to stop here, okay? So each one of these 3-letter words stands for a different amino acid. We call that the genetic code, okay? Now, if you take the 4 bases, okay, and you then come up with all possible combinations of 3 letters for 4 bases, you get like 64 different possibilities, right? But we only have 20 amino acids, okay? So the genetic code is what we call degenerate. Well, what my generation called degenerate. But it just means that you have more than one 3-letter word that means the same amino acid. So you have some redundancy in the code. Okay. You'll notice that in DNA we use A, T, G, and C, but in RNA you will not be seeing any T's. RNA uses the base U, uridine, instead of the thymine, okay? And that has something to do with stability and integrity of the message. And so we will, and resistance to mutation. And we'll talk about that again as we go deep into the structure of nucleic acids. All right, and then, so we get our protein, okay, made from the directions that get stored in the genome. Okay, so a newer feature of biochemistry that was, again, not in existence when I first started studying it, is reflective of our ability, okay, to collect a lot of data, okay, on cells and to process that with computers, okay? So anytime you see the suffix omics, okay, what it means is we are taking all of whatever the prefix is in a cell and looking at it and analyzing the levels of all of those different things. So, all examples of a particular constituent. So what does that mean? Okay, so genomics, okay, is looking at all the genes. So, we have the ability to sequence entire cells, entire organisms. Okay, so we've got the human genome, right? And we can look at the genome from, hundreds of different individuals and compare them and make conclusions about things, whatever our question is, what's the relationship between this gene and, this particular disease. And so we can analyze sequences using computers, right? So if we talk about the transcriptome, okay, what we're talking about are the transcripts, the RNA, okay? So this is one of those cases where we can look at, okay, what genes are being activated when I have this environmental condition or when I have this disease, okay? And there's a difference between what genes are activated, the transcriptome, and the next thing that we call the proteome, okay? So the proteome is all the proteins that get made. So just because a transcript gets made doesn't mean necessarily that it's going to result in a protein being made. There's a lot of steps in that process of going from RNA to protein, a lot of chemical steps, okay? So you have to consider each one individually, okay? So for example, in mammals, okay, RNA gets processed, okay? And if I'm looking at a disease, right, and I only look at the transcriptome, and I say, aha, I've got in this particular disease, I've got, all of these genes, at their normal levels, okay? So if I had let's say I suspected a gene was responsible for it, and it was normal levels of mRNA, I'd say, okay, I can rule that out as the candidate for the problem in this system. But RNA gets processed. So maybe it has something to do with the way that the RNA gets processed so that we don't get protein made. So you have to ask the question, is the protein made? Is the protein made in the form that it is functional? But we can use these different things to investigate, okay, and try to narrow down on where is the problem in this whole cascade of events. Is it a mutation in the genome? Is it the fact that there's something that goes wrong with the regulation of the genome so I don't get the mRNA made? Or does the mRNA get made but not processed so I don't get the protein? Or maybe the mRNA is fine, but one of the enzymes that adds a functional group onto the protein at the end is wrong. Okay, so all of these things, okay, can are important to be able to analyze. And they rely on, so all of these omics things, so I'm going to add another one, okay? So another one is called a metabolome. And remember how we talked about pathways, okay? And so when you're doing metabolomics, what you're doing is you're using mass spectrometry to look at what intermediate molecules are present, right? So this is the collection of pathway intermediates. So sometimes you work, forward, okay, I've analyzed, these 20 people with MS and their family members without, and we've got, we know, we've done genomics, we know there's this mutation, right? Sometimes we do it that way. Sometimes we start from, okay, we know that a person with this type of cancer, their breath smells sweet or whatever, right? And so we can start from that end and say, oh, okay, their cells are making, too much of this particular intermediate. And then we can go back and we can say, okay, what pathways result in that intermediate? Let's investigate the proteome, or let's investigate the transcriptome, okay, in those individuals, and see, where is it that the problem is starting? What's the root of the problem? Okay, so all of the omic technologies, okay, rely heavily on large data sets and computing. Okay, so we collect those data sets through different techniques. So for the metabolome and the proteome, mass spectrometry. For the transcriptome and the genome, it's going to be DNA sequencing and PCR. Okay, so, oops. So all rely on analyzing. Okay, I have moved us from the US to England. Started using S's instead of Z's. Data sets with computation. So the origin of the data differs depending on the type of molecule. So this would be sequencing. This might be real-time PCR. This might be mass spectrometry. This would also be mass spec. Okay, so let's say we've used some of our omics and we have, and we've identified like a protein that is just not functional or has an impaired function in a particular state. We will then, the next step will be to look at the structure, okay, of the normal healthy molecule and the one that has been is the result of a mutation. Okay, so the key concept, okay, for us with all biomolecules is structure determines function. Okay, so that can mean different things, okay, in different cases. Okay, so we can have two molecules like those shown on the left, okay? They have a similar structure, okay? And they have a similar function, right? So we have two molecules, okay? They're from different organisms. One's from a mouse, the other is from a bacteria, okay? The structure looks the same. You'll learn, in this course what the squiggles mean and what the arrows mean, okay? But for now, let's just look at them globally. Do the pictures look similar? Well, we've got a little helix here, okay? There's one here. We've got something, they're roughly the same dimensions and the same shape. They've got things projecting kind of in the same angles. And they have, this represents some metal ions. So one of them has two, but the other one has one. But overall, they look fairly the same. Okay, and you can tell from their names, okay, ribonucleotide reductase, okay, that they perform a similar function. Okay, so with enzymes, the name is going to tell you what the molecule does function-wise. Okay. All right, so this is similar structure, gives me similar function. All right, but now let's look at these two proteins on the right, okay? And so they both have similar structures, okay? They're barrel-like molecules if you looked at them kind of from up above. It's harder to see here. One barrel's a little fatter than the other, okay? But they don't do, but roughly the same, okay? But those two similar scaffolds are used to do different things based on the specifics of the inside of the molecule. So the molecule on the right, okay, is green fluorescent protein, and that glows green, and some of you will work with that this semester. Okay. The other one is a porin channel protein. So this allows ions or water, depending on the porin, to go in and out of a cell, and it acts like a little tunnel through a cell membrane. Okay. So we can have, so structure determines function, okay, but because we have, chemical rules that determine the structures we can make, okay, there is a kind of a limit to the number of different types of structures we can form, and it is advantageous for organisms to reuse common themes. Have you ever heard the phrase, don't reinvent the wheel? All right, so if you think about, I'm an organism, okay, and if you think about evolving over time, okay, every time you have a new function, if every time you did that, you had kind of a completely new structure that had to be coded for by a completely new gene, okay? It's going to take, first of all, longer for that function to evolve. Also, you're going to have a lot more DNA in your nucleus, okay? And so your cell's going to have to get bigger and bigger and bigger. And so there is a level of efficiency that you get out of taking certain structural frameworks, okay, and making variations on them to get different functions, okay? And that is how a lot of, or it's our theory for how a lot of different functions evolved, right? We had something that did this thing, and we had a mutation that enabled it to do something slightly different, and when mutations accumulate, okay, keeping the core structure the same, or the global structure the same, but having small differences in details can give you different functions, right? So, if you look at this building and Chase from the outside, right? They are rectangular brick buildings, right? But if you go on the inside, right? So they're roughly similar structures. If you go on the inside, this building has rooms that are classrooms, just like Chase has rooms that are like classrooms. But then there are some rooms that have fume hoods and more outlets and sinks, and they're designed to be laboratories, right? So we have a similar global structure, but when you look at the interior details, you can see how the functions start to diverge. Okay, so, all right. And so, again, how do we get those differing functions? Okay, is we have our molecules made up of slightly different things. And how does that arise? It arises through mutation. Okay, so proteins, the way they've evolved is that we have, certain mutations that originally happened randomly, and again, but they conferred a survival advantage of some sort on the organism, and so they get propagated. Okay, sometimes a mutation, and when I talk about mutation, okay, I'm talking about when, mutation. It's when you get one or more nucleotides in the DNA that changes, okay? And so in the case shown on the slide, okay, it changes the nature of the amino acid, okay? It changes a negatively charged thing to one that's positively charged. And the slide shows you what the structure of the molecule, how it changes. Okay? And in the case shown here, your mutation, okay, changes the nature of the final protein that gets made to something that no longer performs its function. Okay? So we have a whole bunch of different types of mutations that can occur. Sometimes it doesn't change anything but the DNA sequence, okay? Because, again, of the redundancy of the genetic code. All right, so we are out of time for today, so we'll just kind of quickly wrap up by classifying different types of mutations at the beginning of class on Monday, and then we'll cover section 1 of chapter 2. I've got some practice problems. So chapter 2, we're going to be doing lots of practice problems. So have a great weekend, everyone.